Okay, very good evening, my dear friends. Regarding of CPGET 2023 free online coaching for all competitive exams in plant sciences. Today's our eminent resource person, Dr. P.Manjula Madam Garu, Assistant Professor, Department of Botany, Government, Rbvrr Women's College, Autonomous, Hyderabad. Madam, please welcome and share your screen. Start your session, madam. Please welcome, madam.
Okay, thank you, sir. Sir, is it visible screen?
Yeah, it's visible. If possible, please click on slideshow.
Okay, sir.
కనబడట్లేదు మేడం.
Video on అయిందా sir?
కనబడట్లేదు.
Video on అయిందా sir?
అవును మేడం.
1 minute. Okay.
Slideshow. Please click on slideshow. Okay, madam, proceed.
Okay, so good evening, my dear students. So today's topic is secretory tissues. So this topic is also important for competitive exams. So generally in plants, so along with the simple tissues, permanent tissues, so special type of tissues are also present in the plants. So these are called as the special tissues, or otherwise they are also known as the secretory tissues. So these tissues that generally secrete some fluids which are useful to the plant, or sometimes it may be useful or it may not be useful. So among these secretions, so some are beneficial to the plant and some are not. So these beneficial substances, they are called as the secretions. So cells are tissues generally associated with secretions, hence name secretory tissues. So these are also known as the special tissues. The structure of these secretory tissues also varies. So and the composition of these chemical composition of these secretions also varies from different species, plant species. So the secretions, it may be water, it may be nectar, or it may be salt, tannins, resins, latex, gums, digestive enzymes, hormones, etc. So the secretions, it may be varies in different plant species, and it may be the chemical composition also varies in different plants. So here secretory tissues are again classified into two types based on the position in the plant. So if they are external, so they are called as the external secretory tissues. So the secretions which are internal inside the plant or inside the tissue, so they are called as the internal secretory tissues. So first one is the external secretory tissues. So generally external tissues means they occur external surface of the plant, that is on the epidermis, like that. So they are, it may be unicellular or it may be multicellular. So these external secretory tissues, it may be unicellular or multicellular, so with or without vascular elements. So it may contains vascular elements, that is xylem and phloem, or it may not with the xylem and phloem. So and developed from the epidermis or subepidermal layer. So these external secretory tissues generally they are developed from the epidermis or subepidermal layers. So here the different types of external secretory tissues are there. First one is the glandular trichomes. They are developed from the epidermis, nectaries, and hydatodes. So these are the external secretory tissues. So first one is the glandular trichomes. So these here these trichomes, it may be unicellular, that is it may be made up of only one cell, or it may be multicellular epidermal hairs with some secretory function. So trichomes mean that is the outgrowths and the epidermis, hair-like structures which are present on the epidermal region. So usually have a stalk and head. So these trichomes, glandular trichomes, they contain small stalk and on the tip at the tip region, so small head-like structure is pinhead like. So they are appear like a pinhead which contains small stalk and head. So head is the secretory and head is covered with cuticle. So these head is also covered with the cuticle. So secretion accumulates in the space between the cell wall and cuticle. So these secretions, these are accumulates in between the cell wall and the cuticle. So cuticle burst to release the secretion. So generally cuticle which is present on the outer epidermis. Epidermis is secreted by the cuticle layer. So here on the cuticle, in between the epidermis and cuticle, so these secretions are present and these are released out by the bursting of the cuticle, that is rupture of this cuticle. So here this one is the epidermis. So on the epidermal region, here the stalk cell and here the secretory cell is present. On this here, this one is the cuticle region. So subcuticular oil storage cavity. So when this cuticle is burst, so these secretions will be released out. So next examples for these glandular trichomes are digestive glands of Rbvrr. So generally it is well developed glandular trichome. So generally it is a insectivorous plant, Rbvrr. So here they contains the digestive glands. Generally these Rbvrr, they contains the digestive glands. So they possess the vascular elements and swollen ends of the tentacles are glandular. And peripheral two layers of the cells are glandular and tracheids ends here. So here these digestive glands. So generally these are seen in the Rbvrr. It is the insectivorous plant. So they secrete the proteolytic enzymes. So these enzymes mainly useful for the digesting of the proteins from the. So generally these Rbvrr, they capture the insects. So they are digested by these digestive proteins which are secreted by these digestive glands. So generally, so another one is salt glands of mangrove plants. So generally these mangrove plants in the generally salts are they contain marine water. So here these salts, they helps to remove the salts. Generally they observe the more amount of salts. So these salts will be removed through these salt glands of mangrove. So another example is collectors. So they are the specialized and more complex external secretory structures which are found in some plants. Example, Malus, Rosa and Pyrus. So they found on the young leaf primordia and they produce a sticky fluid which cover the leaf primordia. So these protect the leaf surface. These mainly helps in the protection, and these collectors drop off as a leaf primordia mature. So after the leaf primordia will mature, so these collectors will be fall down. So and provide the protection to the dormant bird. So next one is nectaries. So these are also the external secretory structures. So they secrete the sugary fluid. So these nectar which attracts the insects and helps in the pollination. So again here nectaries are again two types, that is floral nectaries and extra floral nectaries. So floral nectaries means they are associated with the flowers. So generally in the floral parts, they are present in the flower. These are the common type of nectaries, whereas the extra floral nectaries, they are present in the vegetative parts like the petiole, pedicel or stem, and also the and other parts. So and extra floral nectaries are commonly seen in the families like Verbinaceae, Bignoniaceae and Euphorbiaceae. So they are. So the nectaries occur in the epidermis, hypodermis and trichomes. So nectaries have the thick cuticle and this nectar accumulates between the cuticle and cell wall. So this cuticle ruptured and release the nectar. So these are the extra floral nectaries. So nectaries are associated with the vascular structure. So here they are associated with the phloem mainly, and the secretion of the nectar lasts for a short term. So again here Okay. So again here a floral nectaries. So generally they are present in the floral parts. So again, and generally they are present at the base of the ovary. So other than these base of the ovary, so it also present on the other parts of the floral parts, like at the base of the ovary. Example for this is Rutaceae and Brassicaceae, and at the base of the perianth. In ranunculus, these nectaries are present in the base of the perianth, and sometimes it may be present at the base of the stamens, polygonum, and in sometimes it is present on the filaments of the stamens in dianthus. So these floral nectaries, they are present on the dianthus, and sometimes it is present between the stamens and ovary. Example for this is Moringa and Lathyrus. And on the style, in Tarazacum, so they are present on the style. So receptacle of the ovary, Eucalyptus and Callistemum. So in this, so here the floral nectaries again classified into different types. So based on the position, these nectaries are present. So generally they are present at the base of the ovary, but sometimes it may be present at the base of the perianth. It may be present at the base of the stamens, base of the phylum and the filaments and stamens, in between the stamens and ovary, like that. So the position may be varies. So next one is hydathodes. So it is a hydathodes. It is a modified pore, especially on the leaf, so which exudes drops of water. So they release the water. So hydathodes mainly facilitate the guttation. So it is also, so these hydathodes are also known as the water stomata. So generally stomata, they mainly helps in exchange of gases, but here these water stomata mainly helps in release the water. So guttation is the secretion of the droplets of water from the pores of the plants. It is called as a guttation, the process of the release of water here, from the pores of the plant. It is called as guttation. So generally these are seen in grasses mainly. So hydathode is a type of secretory tissue in the leaves. So commonly found in angiosperms. And it is made up of group of living cells with numerous intercellular spaces. So these cells lack chloroplast, and they are also called as the epithem. Another name for these cells are epithem. So hydathodes generally vascular bundles of the leaves ends with epithem. So epithem cells open out into one or more subepidermal chambers and the substomatal chambers, which communicate with the exterior through the pore, that is water stoma. So here also these pores which are present at this outer region. So these pores which facilitate the guttation, that is release of water. So water stomata, which resembles an ordinary stoma in shape and structure, but they are larger than the ordinary stomata. So the difference between these water stomata and stomata are, here water stomata, another name is hydathodes. So it mainly hyda means, so your water, releasing water here. So they do not have opening and closing mechanism like the normal stomata. So these are about the external secretory tissues. So next one is the internal secretory tissues. So these are the secretory tissues which are embedded inside the plants. That is, inside means below the epidermis or in the cortex region or inside internal parts. So store secretory products and maybe single celled or multicellular and sometimes highly specialized. So sometimes these internal secretory tissues, these are specialized tissues. So here internal secretory tissues, they are again classified in different types. That is internal secretory cells and secretory cavities and ducts. And another one is latticifers. So here they are different based on the shape, size, and desecrations. So again they are different types. So again here latticifers, again they are classified into two types: non-articulated latticifers and articulate latticifers. So articulate anastoma, again in articulating articulate anastomizing latticifers and articulate non-anastomizing lattice sphere. So first one is the secretory cells. So these are the specialized secretory cells. So they seen dispensed in the normal cells. So they are present along with these normal cells, and they are also called as a idioblast. Idioblast. So idioblast, it may be isodiametric or elongated tube-like. And most of the idioblasts contain the secretory contents. So they can store tannins, mucilage, oils, crystals. So raphides and crystoliths. So they can store different types of the chemicals. So here generally so the cystoliths. So these cystoliths mainly they contain calcium carbonate. Generally it is seen in Cucurbitaceae, Moraceae. So it is a cellulosic extension of the cell wall in which calcium carbonate is deposited in the form of fine granules in cystoliths. Whereas in raphides, it is calcium oxalates. So these are the needle-like, long, slender crystal lying parallel to each other in the bundle. So these raphides, so these are the raphides. These are the cystoliths. So raphides are needle-like, long, slender crystals lying parallel to each other in bundles, and it is destroyed by the so generally these are seen in alocasia and colocasia and pistia. So these raphides, these are the needle-like, and so generally they store mainly they contain calcium oxalate crystals. So next one is a secretory cavities and ducts. So they are the cavities inside the plant body with secretory contents. So two types of cavities. So based on their formation, that is lysigenous and cygogenous. Lysigenous cavities and cygogenous cavities. So lysigenous means they are formed by the lysis of some cells filled with secretory content. So lysigenous means that is destroying the cells. They are formed by the destroying the cells. So formed by the lysis of some cells filled with the secretory contents and usually irregularly sized and shaped and found in the members of Rutaceae. Lysigenous cavities, these are seen in Rutaceae members. So example for this is oil cavities of citrus and cloves. Next one is the cygogenous cavities. So they are formed by the enlargement of the intercellular spaces between the secretory cells. So they are formed by the separation of cells by the breakdown of the middle lamella. So these cavities are more or less isodiametric, circular in cross section and internally lined with intersecretory cells. So these are called as the epithelial cells. That is, so these cavities which are surrounded by the layer of cells, they are called as the epithelial cells. So these epithelial cells are surrounded by casing cells. So casing cells are thick-walled. So epithelial cells secrete the contents. Generally, these are seen in Ostraceae, coniferales, and Apiaceae. So next one is the lattice spheres. So they are also known as the lattice spheres tissues. So these are the fused tube-like cells filled with fluid called as a latex. So they can occur in any part of the plant body and usually confined to phloem. Here again two types, non-articulated lattice spheres and articulated lattice spheres. So non-articulated means they are also known as the latex cells, which are originated from single cell and elongate by the intrusive growth and occupy intercellular space of the other cells. So maybe or may not be branched. So example for this non-articulated, again unbranched articulated lattice spheres are Euphorbia, Geotropha, Asclepias, and Nerium and Ficus. Whereas the branched non-articulated lattice spheres are seen in Ficus. So articulate lattice pairs means they are also known as a compound lattice pairs. They are originated from the series of cells. So the difference between here is, so non-articulated, they are originated from the single cell. Whereas these articulated lattice pairs, they are originated from the series of cells and the cells are arranged end to end. So common wall may interact or disorganized. So resemble the xylem vessels, hence they are also known as the latex vessels. So again, it is two types, that is articulated anastomizing and articulating non-anastomizing. So first one, that is articulated anastomizing, that is, so lateral connections are established with similar lattice pairs to form a reticulate structure. So example for this is Hevia and Moneyheart. Whereas the articulated non-anastomizing, here there is no lateral connection. So if the lateral connections are called, it is present, so it is called as articulated anastomizing. So here lateral connections are absent, and example here Ipomia, Musa and Convolvulus. So these are the articulated non-anastomizing. So the cell structure of the lattice pairs, so generally the cells contain living protoplast. So vacuoles also well developed, and these vacuoles stores the latex. So cells usually multinucleate. That is, it contains many nuclei, and the latex may be milky white or colored or colorless. So here the latex, it may be in different colors. So milky white, it is seen in Euphorbia and Hevia, whereas colorless in Nerium, and orange or yellowish color in Papaver, this latex color. So the chemical composition of these latex, so generally latex, it is a mixture of organic compounds. They contain carbohydrates, organic acids, alkaloids, terpenes, resins and enzymes. So latex of Euphorbia milli, which contain dumbbell shaped starch grains. Whereas the latex of Papaver somniferum, which contains alkaloids, morphine and codeine. So whereas the latex of Carica papaya, so these are important for mainly for these entrance exams. So morphine and codeine, the alkaloids morphine and codeine are present in latex of Papaver, or Euphorbia, or Carica, etc. So latex of the Carica papaya, which contain digestive enzyme papain, and latex of Acris sapota, which contain chickles used for making the chewing gum. So chewing gum is mainly prepared by using the latex of Acris sapota. So latex of Hevia contains rubber. So rubber particles are suspended in the latex. So when the latex is removed from these plants, the rubber particles clumps tougher. So this is about the secretory tissues or special tissues. So this is also important for competitive exam. So the sources of papain is obtained from the Carica papaya, opium is obtained from Papaver somniferum, chewing gum from the Acris sapota, asafoetida also, root stocks of the Ferula asafoetida. So Indian rubber is manufactured from the latex of Ficus elastica. So this is also important point, and para rubber is manufactured from the latex of Hevia braziliensis. So these are the some of the important points from this. Special tissues are what you call secretory tissues. Sir, so I will stop here.
Okay, madam, thank you very much. Regarding of today's class, have you any doubt, my dear friends? మీరు చెప్పిన తర్వాత డౌట్స్ ఏమి ఉండవు మేడం. రియల్లీ వండర్ఫుల్ presentation. So secretory tissues are very important in the competitive examination point of view. So each and every point is most important. All, so flowering and non-flowering secretory glands and the other osmophores, hydatodes, latticepheric tissue, these are all most important, my dear friends. So this video also we will going to send a very few minutes. So thank you very much, madam. Wonderful presentation. So tomorrow same time we will going to meet in Zoom platform. Thank you.
Okay, thank you, sir.
Right, madam. Right.